Thank you, Simone, and everyone from the Bioconductor team for organizing this conference and for accepting my workshop proposal. It is going to be about human RNA-seq data from recount2 and related packages. Please feel free to tweet it using the BioC 2020 hashtag. Before I start the actual slides, let me just backtrack a little bit and show you some links. So if you're on Pathable, we open the poll section here for questions in case you have any questions. Simone and I are going to try to keep track of your questions. Under files, you can find links to, for example, the package down website or how to run this workshop. So in case you haven't done that, you might want to get that started because it takes a couple minutes to load. But let me see if I can do it myself here. Cool. So this will take a little bit of time to get you an instance ready. Okay. So if you actually want to install everything, it's also possible. If you go to the README file, this Docker command over here that you can copy and paste into your, you already have Docker installed, so I'm going to copy paste it here into my terminal. And then after that, you can go to localhost 8787. And you can also run things on your computer. So you can just get that set up, because it might take a little bit of time to download while I go over some slides. So today I'm going to talk to you a little bit about recount2, the project itself, before showing you any data. So recount2 is a project that we did in 2017, where we uniformly process over 70,000 samples. Right. We uniformly process over 70,000 human RNA-seq samples. I don't know if I selected. Sorry about that. And it's available through the Shiny Apps website. It's a simple table where you can search for terms. For example, here I'm searching for LIBD. I can find a study that of interest that has, in this case, 72 samples. And you can download the gene expression counts. That's what most people use it for. Now, how was this made? It was made using the Rail RNA aligner, written by Abhinav Nilor and colleagues, which is really designed to be scalable and to run on the cloud. And so we ran it on the Amazon AWS cloud because that's the one that we had access to, and it seemed good enough for us. And that's how we were able to process these 70,000 human RNA samples. 10,000 or so of them come from GTEx version 6. About 10,000 of them come from the Cancer Genome Atlas. And then about 50,000 of them come from the Sequence Rate Archive, or SRA. So what do we provide on recount? So we provide the gene counts here, illustrated as boxes in green. But we also provide exon-exon junction counts, here represented by these two big pink bars, pink squares separated by a bar. And we provide big weeks for base pair coverage data that is illustrating this curve. And we also provide exon counts. So all of this can be used for many different things. You can, for example, try to find new expressed regions that are not part of the annotation, illustrated by this orange piece in the curve of the coverage curve that has some reads aligned into it that might be supported by some exon-exon junction reads, and that might be absent from the annotation. But we wanted to make everything really easy to use, and so we borrowed power from Bioconductor, particularly from the range summarized experiment class from the summarized experiment package, illustrated in this purple box that provides 3 different link tables. One of them has gene information shown in blue, that you can access with the row ranges function. Then we have a pink table here of the counts called, you can access it with assays. And then some information about the samples in green with the cold data table. Now, phenotype information is a bit complicated, and we've done some work on it. And so you can actually glue more data, either like using the add predictions function or a new one called add metadata, or you can glue to it your custom metadata. So as a user from R, you can load the recount package. Then you can download a specific study. This here I'm downloading the study ERP001942 from SRA. You can load it into your R using base R functions. And then once you have it, you can start using it. Typically, you want to use the scale counts function from the recount package to start using your data. So this is a lot faster and simpler than having to download the raw FASTQ files for your RNA-seq data, having to process it manually. That means like aligning to the genome, quantifying, all those steps. This is recount2. Recount1 is from 2011. Mike Love, the author of DSeq2 and other R packages, he found the first version of recount really useful for developing methods. And so we wanted something that could be used by downstream biological users, but also power the development of new methods. And so over the years, there's been a couple of recount2 related projects. Some of them I'm going to mention them here, and there's a lot of them. This is just a little list, and you can see there's a lot of text. So this next slide just highlights a little bit of what the type of projects that we've done. Some of them are about documentation, like the one that this workshop is based on. Another one is based on phenotype prediction, transcript quantification, applying the data from recount2 to proteomics, looking at other annotations or custom regions. This one in particular I'm really happy about because it involved people from Mexico, from my country, where maybe not necessarily everyone has access to high computing power or storage space. And so in a way, recount2 is a project that democratizes the access to the data. There's also other studies done about validating data from others using the data from recount2. And like I said, also the development of new methods, which is what recount1 mainly was useful for. Another related project from our team is Snaptron, which allows accessing the exon-exon junction data from the recount2 project. And as of 2020 in BioC 3.11, there's a new package called SnapCount for our users of Snaptron. One of the projects that I mentioned was about phenotype data. This was led by Shannon Ellis, a postdoc in Jeff Leake's lab at the time. And this project was really useful because we have a lot of data from recount2, a lot of gene expression data in different formats, so the gene, exon, junctions, express regions across many samples, but how can you actually use it to answer questions in biology? Well, you need to know what those samples are what, like, for example, their sex or their age or their diagnosis status. And so just getting sex can be tricky. Here Shannon tried, did a lot of work, and like out of the 50,000 samples for SRA, she found information for only 3,600 samples. That's because when people upload data to SRA, it's a free text form. Everyone does it very differently, and there's no metadata requirements really. And so using GTEx and TCGA, which are two large projects with a lot of good metadata, she was able to build a predictor, test it, and then apply it to SRA. One of them here, for example, is sex, which was very good for GTEx and TCGA. For SRA, it's not as good as you might expect, like around 90%. But that's also because the ground truth is a bit messy, and some of the samples are mixed samples too. For tissue, it gets a little bit tricky because it turns out that predicting healthy tissue, because the predictor is based on GTEx, which are healthy individuals or controlled individuals, works quite well with TCGA, but like on the healthy tissue, but not so well on the cancer tissue due to several reasons. And then on SRA, the ground truth here that we had was very limited. So, and it was also, it wasn't really the truth. It was based on another algorithm. So that's why these 50% seems quite low. But now you can use that using the add predictions function. You can just add that those results. And that can potentially help you use the data more. And one such project that we did is recount brain, where we looked at all the brain predictions because it was the second highest predicted tissue in recount2. And we did a separate approach where Ashkan Resmara, an MPH student working with me at the time, he manually downloaded a bunch of papers and read the papers and manually constructed a table that then our collaborators, Sean Davis, Dustin Sokołowski, and Michael Wilson helped us expand in different ways. And now you have a little table here, a curated table with information about a subset of the studies. This was only really 62 SRA studies. recount2 has 2,040 almost studies. So it's a very small subset of them, but this is kind of what you need to do in order to actually use the data. So we made a lot of effort to make the processing of the data and the availability of the data simple, but then the metadata is really sometimes an Achilles heel because of how people share the data. So now with recount brain, there's this, we added this function called add_metadata that you can use it to download the full recount2 table, explore it, find your study of interest, and then you can go back to using download study from recount, download it, add a metadata, and then start using it for different like types of analysis. So that's a lot of the work that was done by different people here for the recount brain project in particular. These are people I'm showing in this slide. So that hopefully gives you an overview of what recount2 is. And now you might be interested in actually learning how to use it. I don't see it, it's not refreshing on my screen, sorry.
Well, it had to do with the existence, with the possibility of such a resource for epigenetic data like methylation. I think that's
Oh, okay. Okay, so methylation. So there has been interest by some of our collaborators in doing a recount for methylation data. And I see now there's another question about, is recount2 a snapshot at a certain point and it's growing over time? So I have a slide near the end about that, but the quick answer is yes, it is a snapshot. It wasn't intended as a snapshot, but it became a snapshot. So I'll have more on that later. Thank you for your help, Vince. Okay, so let's actually look at a bit about how to use the data. So there's some of you might be more visual, so I'm going to use the slides. Some of you might prefer the text. And so actually, if you go to the precompile website, a lot of what I want to explain is written in text format here, in case you prefer to either read it while I'm talking or read it later. Right. So how do you actually use the data? Well, I mentioned that you need to use the recount package. And in particular, there's two functions, download study and scale counts, that are the most useful functions if you want to actually use the data. Now, scale counts is a bit complicated, which is why I'm going to have to spend a bit of time explaining what it is. So let me use some cartoon images here. I'll make this a bit bigger. So imagine that we have 16 RNA-seq reads shown as these pink boxes. Each of them are 3 base pair longs. That's why they have 3 squares. And we have a reference genome of about 16 base pairs. So the first part in RNA-seq data is that you need to align the data. You need to find what part of the reference genome these reads come from. And so in our case, we use the RNA aligner, and that can give you some output like this, where you can find that, you know, this read over here aligns to the first 3 bases. Some of them might be split because maybe they span an exon-exon junction. Some of them might be cut. They might be trimmed a little bit. So these are represented by these purple boxes. We only like a portion of the read shown. So once we have that, I mean, recount2 provides that type of information, but we need to build the gene counts and the exon counts that we're providing to users. And so we're actually going to go back to the coverage data before we can build this range summarized experiment objects, and in particular the counts, the pink table that has information. So let's imagine now we have an annotation of interest that tells us the coordinates of specific genes and their exons. So in here I'm looking at a single gene from an annotation that has 2 isoforms that involve a total of 3 distinct exons. Isoform 1 uses a long exon and then a short one at the end. Isoform 2 uses a short exon at the beginning and then the same exon 2 at the end. We can identify the disjoint exon pieces for a given gene. So these are the different segments of expression. And once we have those, we can go back to our line data, and for every base, we can ask how many reads overlap that base. So for this particular first base, there's 3 reads. The next one is also 3. The third base is 5 reads, 1, 2, 3, 4, and 5, etc. So once we do that, for every disjoint exon, we can compute the sum of the base pair coverage. So this is 19, 7, and 10 in this example. And we can then compute the overall gene expression base pair coverage, which is the sum of the disjoint exons. So 19 plus 7 plus 10, that gives you 36. So this is a very different way of counting compared to the typical RNA-seq data processing framework. And we're counting base pair coverage data. We're not really counting reads. So one way that you can transform these base pair coverage counts is to divide them by a given read length and then multiply them by a target library size divided by what was the actual number of map reads. And that can give you scale read counts. Then that's what the scale counts function is doing. Now, one way of doing it is you can actually sum the area under the coverage, AUC, which is the total sum of the base pair coverage across the whole genome. So in this case, along our 16 bases of reference, that ends up being 45. And so you can represent these numbers 3, 3, 5, 4 as a little curve, 3, 3, 5, 4, 4, etc. Right? So area under coverage is kind of like area under the curve of this coverage curve. And so the math turns out that the number of map reads multiplied by the read length gives you the area under the coverage. So we have our base pair coverage counts here. We can also easily compute the area under the coverage from the bibwig files. And so multiply by the target library size, that's how we get our scale read counts. And so that is a bit tricky and different from what other RNA-seq pipelines do. And that's why that's the main source of confusion and questions that I get. Now, in this particular image, we haven't used all the data. For example, we didn't use these two, we didn't use the information over here or the exon-exon junctions. And you can actually write new methods to try to use that information to find, for example, a potential third isoform, which might be supported by like this split rate over here that has an exon-exon junction, some data here, maybe this reading that is grayed out that also supports this third base there. That's potentially something you could do. You could just look at the reads that have some coverage, that maybe don't have exon-exon junctions supporting them. Or you might also find reads that tell you that maybe the end of a particular exon is not correct, that maybe you need to have, add some bases to the annotation. So you can imagine a lot of ways of improving the known annotation and exploring what's going on. And the nice thing about this is that because we have processed 70,000 samples, you can ask later if this is something that is rare or this is something that happens in other tissues, in other studies, in other type of diseases, etc. So that's the potential behind recount2 and what you can do with it. So one particular example of looking at unannotated expression is from using a method I developed called DERFinder, which is also a Bioconductor package, which identifies express regions for you using this coverage curve shown here in the first panel. Once you find the coordinates of those express regions, you can then quantify them and do your typical differential expression analysis. And in particular, we applied it in 2015 in this paper of 72 samples, which is available from Recount. And this is the one that is used on the Recount workflow per se, and not on the Recount workshop that I want to show you today, because that one is too big for us to use in our computers right now. And so using that data set of 72 samples, we found a lot of differentially expressed regions that were preferentially expressed in fetal samples over the other 5 age groups. And so these are some of the methods that we developed prior to recount2. And we wanted to be able to continue doing this type of analysis. That's why recount2 powers this type of work. And then in this particular study, we used the BrainSpan as an external data set to validate the effects that we were seeing. So that used DERFinder, which has been available since 2014 in Recount. I couldn't remember what's the BioC version for that when I was making this slide. And then something new is the GenomicState package, which I'll mention on the code side. It's been around since October 2019. And really this is in response. The idea of this comes from one of the reviewers for the Recount workflow was libdRSE, and he gave an idea back in 2017 that it took a couple years to then implement. Not because it was hard, just because I didn't spend the time on it. Sorry about that. So that's how you can use Recount. We have a lot of examples on the web, and but because it's a little bit confusing, that's why I wanted to dive a bit more into the code, into the scale count side before I show you more of the code of how to do this. Now, this is really a team work and involves many labs at Hopkins. And some of the people are no longer at Hopkins, but they're still part of our recount2 family. And everyone has different grants and multiple positions available. So if you're interested in continuing to build recount2 or related tools, please let any of us know. There's different aspects of it that each of us is working on. And that's also why recount2 was maybe challenging to first carry out, because there was no single grant that could pay for all the competing costs related to it. And that's also why, related to that question about the snapshot, maybe why it became a snapshot. It wasn't intended as that. Right.
There are, Leo, there are a couple of more questions now. I don't know if this is perhaps a good timing to reply to some of them.
Yeah, sure. So let's see. Questions for me. I did see a question about scale counts. How does it work? I explained some of the math already, but in reality, it uses information that is stored in a range summarized experiment object that is very specific to the recount2 objects that we made. And so that's how it's able to access both the area under the coverage and the counts. So it's really is like a small transformation, and it updates the assays counts slot. You didn't refresh.
Do you want me to read the other? I can read them for you,
Yeah,
if
sure.
you want.
Thanks.
Right, and there is the next one is it's recount to a snapshot at a certain point.
Oh, that one
in time.
I did.
Yeah. You did, right?
Yeah.
Then okay, then did you see the recount package is a fantastic resource, etc.
Inhibition for a CheapSeq resource. CheapSeq. I would say that, I mean, even though there is some interest in CheapSeq from our side, we don't have the bandwidth to do a CheapSeq resource for now. So I think, I mean, we could definitely have a meeting and share with you some of the stuff we've done for RNA-seq. Maybe that can help you get started on your CheapSeq project, but I don't think we have the bandwidth for that. So basically, we're interested in collaborating, but we don't have the power to do it. Thanks. So let me go to our code. So I'm gonna use the cloud one. That this is thanks to Sean Davis. Our studio is the username. Welcome -to -BioC 2020 is the password. I want to remember that. So the code itself, there's a couple ways you can access it. I'm going to use this vignette command with under quotes recount -workshop, and then also comma, and then under quotes recount workshop with capital W 2020. There's other commands you can use. But what this does is it opens under the help section the vignette. And I'm doing this specifically because I don't want to copy paste code. So I'm just hiding some of the panels from our studio. And let me increase the font size. All right. Oh. So we already did a lot of the introduction. So I'm going to jump into gene level analysis. All right. So the whole code here takes 7 minutes to run if you run it fast. But I mean, you're more than welcome to try it out later. And there's several venues you can use for asking questions that I'll try to monitor and get back in touch. So actually, I'm going to scroll up, sorry. There's a piece of code here where I load all the related packages here. So what I'm going to do for this whole dynamic is that I'm going to select code under the help page. And then you can like manually right-click copy, then right-click paste. But that would take quite a bit of time. Our studio, though, has this feature that if you select code here, if you're on a Windows computer, you can use Control-Enter. If you're on a Mac computer, you can use Command-Enter. And that will like automatically evaluate whatever lines of code that you have highlighted on your console. That way you don't have to copy paste anything. So I'm loading all the packages because it takes a little bit of time to load all these packages. And then I'm gonna skip to the actual gene level analysis. So section 2.3 of the vignette. So this particular package called the Recount Workshop 2020 package has data for a study called SRP056604 that I chose because it's small and because it's also present in Recount Brain. And the data is already provided inside the package. So if you're using Docker, you'll have all the data there. If you install it from GitHub, you can also have the data there. So we first need to find the data, and I'm going to use a system file function from base R for that. And so that should look something like this, which is the location of our library in our Docker image, Recount Workshop 2020, then external data or ext data, and then our ID. Boom. So let's say you actually wanted to use recount. You would first load the recount package, which I mean we already did. And there's a function there called abstract search, which is a very simple search function. It only provides grep syntax, nothing. It doesn't do like nothing fancy for searching, and it's based on some abstracts that we have for the 2040 studies. So here I'm really searching for a very specific keyword already, hippocampi of 4. So that's why it's only returning a single study that has 8 samples. It's from human, and the project ID is the one that we're going to use. But that's one way you can use. Another way is to use the website, the Recount Shiny website. So now the data, like I said, is already available. So you can just load it, but using the load function. But in case you were to do this outside of this type of environment, you will need to use the download study function to access the data from the web. So, well, we already have the data. We can check that the data exists where it's expected to be, and then we can load it. And I'd like to use the verbose true argument for load such that it shows the name of the objects that we're loading. Once we loaded the data, this is a range summarized experiment object. So the metadata, the information about the samples, is stored in the call data slot of the object. Now, we have 8 samples total. That's the number of rows. And we have 21 columns, which are the basic 21 columns provided in recount2 for any data set. Let's look at the column names of those. And so it's like the project ID, sample, experiment, run IDs. But then also some information such as like the read count reported by SRA, the number of reads downloaded, the bigwig files, and some other information, the AUC, which is that area under the covers that the scale count functions uses. So this is very specific now to recount2 in that we expect to have these 21 columns all the time. Now, we have also this function all metadata, which can also be used for finding studies of interest. And what it does is that it downloads the metadata for all the SRA samples that we have, or the GTEx or the TCGA samples. So you can see here is actually downloading a file called metadata clean SRA. And so I'm just showing you that because I wanted to show you, for example, a sample here that has exactly the same number of reads downloaded by SRA as the ones reported by SRA. And overall, we can see like for this study 045638, what is a summary of the proportion of the reads downloaded by SRA? And ideally, you want it to be 1, but in some cases it can be low. So here there's actually a sample that we can only download around 57% of the data. And so this happens because RNA was run on the cloud, and the data source sometimes can have connection problems. And even though we have a lot of checks and retries, eventually sometimes we need to give up on a particular sample. So we try our best, and sometimes our best is only 57%. We can look at these big numbers that are like the sum of the base pair coverage. So this is like 14 trillion using the AUC. We can look at, for example, the number of map reads per se. whether they were paired end or not, and the average read length for them. If you multiply the map read length and the average read length, you get a number very close to the AUC in practice. There's also information that we have about the samples. One of them is from the Shark beta project, which at the time they were working on publishing, but I think by now they have abandoned this project, or not being able to continue. And the Shark project, what they did is they looked at the abstracts of papers and inferred tissue from the abstracts and other information. And so for some studies, like the one we're using, we don't have any tissue predictions. For other ones, like the example data set in the recount2 package we do, which is the blood. These are samples from blood. Now, we tried to provide more information, and if you attended the first workshop of this BioC 2020 conference, Sean Davis mentioned the GeoQuery package, which is a package he wrote for accessing information from the Geo database. And so we actually use that GeoQuery package, and we're able to obtain the geo accession information, the title as provided by the authors of the data, and what's called the geo characteristics. So this geo characteristics is kind of like the Wild Wild West. And that's because that's information the authors provide. And they can provide it as key value pairs, but these are text-based key value pairs. They can write them whatever they want, and that means that sometimes they can make typos or choose to use different ways of describing the same information. So for example, let's look at the first two titles in the example data set from the recount2 package. And so really here, the title of the samples is very complete. It describes what cells they are, whether they were cultured with or without doxycycline, and for how long. Now that information they might also provide it on the characteristics of these key value pairs. So for example, here they say cells, this is K562 with shRNA expression with no treatment or with treatment. And then they can have details about the treatment. One of them is just puromycin. The other one also has doxycycline for that particular study. But now, not always the same wording is used for describing everything. Because for example, here, if we look at the very first sample, we see cells K562. If we look at the 11th sample, we see cell line K562. It's the same information, but like if you're writing some code that will use the characteristics, you need to be able to recognize that these two are the same. And so we basically provide this Wild Wild West as is to the users. It's hard for us to try to clean it anymore. And try to make it simpler. Now, if you go look at the particular study that we're working on, we have two characteristics here. Let's look at, we have 5 key value pairs. Let's look at the first 2 samples. And so we can see that there's age, gender, Braak stage. That's because we're talking about Alzheimer load, what's it called? I think it's Alzheimer's. Yeah, I think it's Alzheimer's, but it might be the other one. Sorry. We have what control group it is, the APOE genotype, and what tissue it is. So once we see the structure of all of this information for all the samples, we can write some code to then try to infer what is a load sample or a control sample, what is female or male, and then like extract the years from this string over here, or like the greater than 90, we're just gonna say that it's 90 years old. So that's something we can do manually. So this is in that diagram that I have of the range summarized experiment, this is really like the brown box where we're adding some custom metadata. So we can see, like, for example, how the cases and sexes are balanced. And they seem to be okay balanced for like a small data set over here, or the age and case and control status. And it's a bit mixed, so that's good. It doesn't mean like, if you were at Chloe's presentation, we want to avoid some big compounding effects. So like if age and case control status were perfectly confounded, that would make the analysis harder. We can also add to our object the predictions from Shannon Ellis, the phenotype predictions. And so that involves some new columns over here in our data set. That includes the reported sex, predicted sex, which is male or female, the predicted tissue, which in this case is brain. That's how actually I selected the samples to work with. Now, some issue that we can observe here is that if we compare the observed sex versus the predicted sex, we have some mismatches. We have 4 predictions, mismatches, and we can try to like look at other covariates, other phenotype information that we have, and see if there's anything there. But it doesn't seem that whether the sex is matching or not is related to the case control, for example, or the age. It's in the middle age groups. So this is something where, like, luckily we do have reported sex, so we could use that instead of the predicted sex if we believe that over the predicted sex. Or like the number of map reads, for example, between the matching sex and not matching sex. That opened a plot on my RStudio window. I need to go back to the help side, continuing the vignette. Now, something else you can use is the SRA run table. So you can actually download this. The format of this table has changed over time. It used to be a tab delimited file. Now it's actually a CSV file, but they still use the former file extension .txt, which is confusing, instead of .csv. So that file is already provided here. If you're using the 2019 or 2017 version of this workshop, the file is actually the former version of the file. So the code that is different between the two. So we can read now that information with like the read CSV function. There's more information here that is, some of it is already overlapping with information we have from Geo. Some of it might be different. And this is really study to study dependent. I like to add the SRA underscore extension to the column names, just so I know that this information came from SRA. And so I'll do that right here. Now, the information in that table and the information we have might be in different order. So I'm going to use a run ID from both tables to match the information. And we can make sure that it's actually in the correct order using the identical function. It should be, because I just ran that code and I missed 2 parentheses in my command to run it. And so you can now append the data using cbind. And now that we have all the data, we can actually try to move and start analyzing. There's also create data for brains, which is this recount brain project that I mentioned. You can like use the add metadata function to download that, find the particular study, which is, this was chosen for that because it's a study that is present in recount brain, and from it we're gonna use information such as the RIN, PMI, which is post-mortem interval, the units of the PMI, disease status. So this is created data for this particular study, which unfortunately is not available for every single of the 2040 studies in recount2. Although if you create data for any tissues, we will be more than happy to distribute it through the same functionality. So now that's how you can check recount2, or you can just add the data using the add underscore metadata. function. And now we have 104 columns of phenotype information in our data. Next, we can do the differential expression analysis. There's a lot of bioconductor packages that specialize in this process. And so there's different ways of doing it. I'm going to use the scale counts function. And just to highlight that I'm using a different type of object, I'm going to rename it to underscore scale. And I'm going to delete the first object. You don't really need to do this. You can rename, you can overwrite the object. But I just want to highlight that the data has been scaled now. So here I'm just going to use some template DESeq2 code for analyzing the data. It has to be integer counts. And so we actually rounded the data to zero decimals to actually use counts and to provide it something that it knows how to handle. That is designed to handle. And I'm going to run a simple model that has sex, age, and PMI. And this takes a minute, not a minute, but it takes a couple seconds to run. And once it's done, we can make a MA plot showing the results. So I'm running a little bit low on time. So I'm going to just show you some of the stuff you can do just by browsing the vignette. Once you have the results, you can use these region report package for making an interactive report of the differential expression data. And you can use other bioconductor packages, for example, for gene ontology enrichment analysis, such as cluster profiler. So that's what you can do at the gene level. You can do other models. This is one other model. And just like we did the gene analysis, you can also do an exon analysis. So here we could download the data. And I'm going to actually bypass a lot of the steps with it by just pasting into the gene level data. Sorry, that's the MA plot we were doing. I'm going to paste into the gene level data, the exon, into the exon level data, the gene metadata that we did. And so you can run the same type of model at the exon level, get your results. And you can compare, for example, which are the genes that have differentially expressed exons versus those that are differentially expressed at the gene level themselves. There's different reasons why that can happen. And they're highlighted in a paper by Son et al. In 2015 in F1000. So that's how you can do some of this expression level analysis. You can also do base pair resolution types of analysis. So there's a function in recount called express regions that uses DERFinder behind the hood for defining those express regions. Here I'm running it with like some default parameters. In this case, it gives me 45,000 regions for this data set for chromosome 12. So that's a lot of regions. And so a lot of them might be really small. It might be just one base pair long. So here we're going to drop everything that's below anything that's below 60 base pairs long. And that gives us 22,000 regions to work with. Now, in order to actually use these express regions, right now we only have the coordinates of the genome. We can use the coverage matrix function to actually compute the base pair coverage in a similar fashion to how we did the full gene counts. And so this takes, it used to take 45 or so seconds, and now it takes like 28 or so. When I ran it earlier, it ran in 26 using this cloud environment. So once you have that, you can then also do your like DESeq analysis, find differentially expressed regions, kind of like what we were doing before. Make your volcano plot, all of that.
Leo,
yeah.
You have two more minutes only.
Yep, thanks.
Sorry.
Now you have our regions. If you actually want to visualize the regions, we have the BigView files that on the web available through this Duffel website. So with those regions and using functions from DERFinder, you can visualize the data. Now to make all this process simpler, that's why we made the GenomicState package that has a lot of the data that you need for making those plots. So I'll skip a little bit of that. And you end up with plots like this that show like the base pair coverage, which are, these are some example regions. They're not the prettiest regions that have data, they might overlap express features. And that's the end of that code there for the workflow, for the workshop and workflow. Let me go back to, yeah. Well, why is it hiding? Oh, it's on my second screen, sorry. So that's a code that you can go through in more detail. And I just wanted to end with some issues that related to some of the questions earlier. So there are some issues with recount2. Rail RNA is hard to run outside of a Hadoop cluster, which makes it hard to have major updates. And it's only human data. So a lot of people ask, where's my favorite data set? And the answer is like, oh, it's not there, sorry. It requires a lot of manual post-alignment work to actually create the resource. And it uses one version of the GenCode annotation, GenCode version 25. There's also a bit of a problem with overlapping genes because we might count the same bases twice or more. And so that can lead to some issues in some analysis. So the future is really recount3. That's why on Twitter I said that this might be my last time having a recount2 workshop. We're going to use a different aligner. It's going to have mouse and human data with studies and collections. And it's going to be hopefully auto-updated with several annotation choices and potentially faster tools for re-annotation quantification. Also, it's going to change, the inside of it is going to change a lot in how the files are provided. And so hopefully it will come to your nearest Bioconductor mirror in 2020, kind of like a movie. And so that's it. And if you have any more questions, please post them publicly over instead of email, because that helps others. The recount package, the recount2 feature requests for, the support website and the issues for asking questions or making feature requests. And I know this is really confusing and I'm sorry about it, but there's a recount project from 2011 that has no R package. The recount2 project, that the R package is called recount, not 2, because we were not allowed to have a 2 given Bioconductor rules. And now we're going to have the recount3 project, and to try to solve the confusion, we're going to hopefully rename the package recount3 instead of recount2. Otherwise, that would be even more confusing. And so that's it. Thank you very much.